Welcome back, everybody. Hope you had enough time for a small comfort break. My name is Selena Sim. I'm a Macmillan Consultant Respiratory Physician at the Queen Elizabeth University Hospital in Glasgow. And I'm delighted to chair this final session of what's been a fantastic day. I think you can agree. And this session is on a research showcase. We have two fantastic speakers. The first one is Professor Daniel Murphy from the Institute of Cancer Sciences at the Beatson Institute, who is got a specialist interest in genetically engineered mouse models and in mesothelioma. Thank you, Daniel.
Thank you very much, Selena, and thank you, Kevin, and all the organisers for putting this together. This has been a fantastically informative day and very, very helpful for me. I think very helpful in concentrating our research focus. I'm going to apologise up front because this talk will get quite molecular, and on top of that, I'm going to show some histology images that may be upsetting. So if it's not your cup of tea, feel free to turn off the screen. So starting off with what my lab does, we use genetically engineered mouse models to try to model how the disease develops, how mesothelioma develops over time from exposure, incorporating loss of the same or mutation of the same genes that are mutated in human mesothelioma, and to try to track the entire course of what's happening over time as the disease eventually reaches the point where we have to euthanize the animal. Now, what does it actually mean to use genetically engineered mouse models? And I thought I'd really get into to try to demystify some of the things that we do. And so what we have are tools that enable us to control gene manipulation, whether a gene is expressed or not. And we do this, for instance, here, showing you an example of how we induce gene deletion or loss of a gene. And we do that by inserting these triangular elements called LOXP sites within the gene. And then we expose that gene to an enzyme called recombinase. And upon exposure to recombinase, the intervening sequence between these two triangles gets cleaved out, essentially deleting a chunk of the gene. And this is analogous to what happens in the genome of humans exposed to mesothelioma or to asbestos, where you get recombination events and loss of big chunks of DNA in the genome. We're also able to induce gene expression artificially, and this we do through genetically engineering in stop elements that can prevent normal gene expression. So we have a stop element, again, flanked by two of these triangular LOXP regions. And in this instance, upon exposure to recombinase, we get rid of the stop element, enabling a silent gene that we've engineered in to not be expressed to now be expressed in a controlled manner and in a way that we can time when it gets turned on. And so in thinking about how to construct a good mouse model of malignant pleural mesothelioma, we incorporated three elements in our thinking to guide how to generate these models. And first and foremost, we felt that it was crucial to be informed by the human mutation data. And so in recent years, we have seen a couple of really comprehensive studies, really elegant studies that have looked at the deep molecular features of human mesothelioma and identified the mutations that occur most frequently in human patients. And I'm showing you the four most frequently mutated genes here, including BAP1, CDKN2A, NF2, and TP53. And the mouse modeling community has already generated a mice where we can controllably delete each of these individual tumor suppressors. And what we do in my lab is that we've interbred multiple mice such that we can now delete combinations of up to 3 of these tumor suppressors in a single go. And that enables us to really model very aggressive human mesothelioma. The next element that we wish to include is that we only want to mutate these genes in the appropriate tissue. In this instance, this is within the mesothelial cells of the pleural space. And we achieve this through viral delivery. We use recombinant viruses to deliver the recombinase enzyme into the pleural space. The viruses penetrate into the mesothelial cells, and the recombinase is then expressed within those cells and introduce the mutations in a controlled manner. And then the final step is, I think, very importantly, we also inject asbestos into the pleural cavities of these mice, so that we are in fact having that asbestos-driven chronic inflammation that is part and parcel of how mesothelioma develops. And I think this is a key element that I think has been overlooked by some of the genetic purists who in the past would have thought, well, if you can get the disease with the genes alone, then there's no need to be messing around with asbestos. That is indeed a quote from one of my colleagues in the past. I feel that having the asbestos in our mouse model enables us to realistically model that chronically inflamed state, and that inflamed state doesn't go away once the mutations arise. That inflamed state remains present in all the patients for the duration of their lifespan. I should have given you an alert here that there was distressing images coming. This is looking down at the level of individual tissues in mice where we've either injected asbestos alone, or we've injected a combination of asbestos and our Cre-expressing virus, and we've knocked out 3 tumor suppressor genes. And the tissues that I'm showing you on the left all show the typical disease that you would get, a sort of an inflammatory response that you see within the mesothelium upon exposure to asbestos. And this is the degree of inflammation that you get within 3 to 4 months within our mouse model after exposure to asbestos. At the same time, if you look on the images on the right-hand side, when we delete 3 tumor suppressors in the presence of asbestos, we get this really aggressive mesothelioma disease that has caused us to actually have to euthanize our animals. I think you can appreciate here that the black arrows are pointing to invading mesothelial cells that are invading into the underlying musculature, into the diaphragm, into the surface of the lung, and also into the heart. And it really gives us, a clear indication of just how aggressive and destructive this cancer is. We have looked at survival, analogous to what is done with human patients. We also do survival analysis in our mice, and I think this really drives home the point of the importance of including the asbestos in our mouse model, that in 2 instances here where we have genetically deleted 3 tumor suppressor genes that recur in human cancer, different combinations of tumor suppressor genes. When we include asbestos in the red lines shown you in both of these graphs, when we include asbestos in these cohorts, we really accelerate disease onset, and the speed with which these mice succumb to mesothelioma and have to be euthanized is really quite remarkable. But I think it's very reproducible, and really what it gives us is a model system now where we can develop full-blown mesothelioma generate full-blown mesothelioma within our mouse models within about 100 days or 120 days. I think it's an important point that our model systems show the same characteristics and the same spectrum of disease that is seen in human patients. So depending on which combination of genes we mutate, we either get mesothelioma in the mice that is predominantly sarcomatoid, which is the most aggressive subtype of mesothelioma, or indeed using a different genetic combination, we get predominantly epithelioid disease, and patients with epithelioid disease do survive much better than patients with sarcomatoid disease for reasons that we still don't fully understand. And really, when we go deeper into our mouse models and look at the molecular features, so what I'm showing you here is, some of you will recognize this as a heat map. Some of you that won't make any, it won't be familiar to you what a heat map is. Essentially, this is a way of looking across a whole spectrum of molecular features of each individual cancer. And so each one of these columns, each one of these vertical columns represents one cancer, and we're looking at the molecular characteristics of different pathways. And really, this is a way of grouping together cancers that are similar in their molecular features and differentiating them from cancers that are different. I think one of the striking features of this analysis, it tells us about the molecular heterogeneity of mesothelioma is really, really profound. This isn't 1 or 2 or 3 simple diseases. This is really a spectrum of diseases with very, very different molecular features. And I think that's one of the reasons it's been very difficult for clinicians to hit upon one successful treatment strategy to advance this disease. But if we take these human samples and we mix in our mouse samples and ask, really, how well do our mouse models recapitulate human disease? it's really reassuring for us as molecular biologists to see that the mouse samples scatter out amongst the human samples. They're essentially indistinguishable from the human samples, which is it's actually quite surprising. You would think that, mice being mice and humans being humans, that the mouse tumors would all separate off and be completely separate from the human tumors, and that's not what we're seeing. So it really tells us that the molecular characteristics of the mouse tumors that we're generating really do resemble the spectrum of heterogeneity that's being picked up in human mesothelioma. And so, just to touch upon some of the themes that we've hit upon earlier in the day, one of the things that we want to do with our mouse models is to de-risk new treatments and to explore using new combinations of drugs or new drugs that look promising, to put them into our mouse models and see, is there actually any therapeutic benefit of testing these drug combinations and sparing those processes from actually being performed in patients. So let's get some idea of the toxicity. Let's get some idea of the efficacy of these drugs before we go into the clinic and treat patients with these. And so this was a publication that we published just a few months ago, actually, in collaboration with Marion McFarlane and Anne Willis from the MRC Toxicology Unit in Leicester. And they had prior data looking at rates of proliferation in resected tumors, and could separate out, the tumors with very high rates of proliferation here scored as high Ki67 versus patients with lower Ki67. This is a marker of proliferation. That the patients with higher proliferation did worse than, or the patients whose tumors had higher rates of proliferation did worse than the patients with lower rates of proliferation. And that seems to be obvious, but it wasn't necessarily something. It wasn't necessarily a given. And what they found was that the rate of proliferation or the frequency of proliferation within these tumors correlated very well with the activity of one particular molecular feature called mTORC1. And the mTORC1 pathway controls the rate at which proteins are produced within cells. And so this suggested to us a specific pathway for which there are drugs available that could be modulated and could be targeted to perhaps slow down proliferation, and that might have a therapeutic impact. So the actual molecular pathway here, this is Greek to most people, but what we hit upon here was a druggable pathway centered around PI3 kinase and feeding down into mTORC1. And we used a drug combination from AstraZeneca, AZD8186 and AZD2014. And when we took this drug combination and tested it in our mouse models, we did indeed find that there was an extension of survival in our animals upon treatment with this drug combination. Now, as you can see, the red line here isn't that far from the blue line. So, this again is a modest improvement. But I think one of the points that gets missed in some of these analyses is that this combination of drugs was tolerated much better by our mice than was a combination of standard of care chemotherapy. So we've tested cisplatin with pemetrexed in our mouse models, and the mice really suffer when we treat with standard of care chemotherapy. So we've struggled to actually build good cohorts of animals that last long enough to give us meaningful data using chemotherapy. So I think really one of the things that we're hoping to see from testing these drug combinations in our mouse models is that we see kinder, gentler therapies that, although they're clearly not curative, but they may give the patients an easier ride through the clinic. Turning our attention now to, what are some of the additional benefits of using genetically engineered mouse models? I think we've briefly touched upon the latency period between exposure of humans between exposure and the initial presentation with symptomatic disease. And it's not uncommon for patients to have been exposed to asbestos 30 or 40 years before they develop symptoms that cause them to present to the clinic. And when they do present to the clinic, typically, patients will go in with potentially pleural effusion, fluid in the lungs, but very often they're initially diagnosed with potentially benign disease where there's no evidence of invasion, and only about 12 to 15% of those patients that initially present will actually progress to be diagnosed with mesothelioma. Now, the problem we're faced with right now is that in the clinic, we're unable to predict who that 12 to 15% are going to be. And if we knew who they were, then we could potentially start intervening earlier and start treating their cancers earlier. So one of the big advantages that the mouse models give us, I've listed some of the points here, using our ability to control the genetic characteristics of our mice, we can determine the outcome essentially. We can genetically determine whether a mouse is going to get mesothelioma or indeed not. And that ability to determine that a mouse is destined to get mesothelioma enables us to look back over time and to sample, those genotypes of animals over the entire period of disease development, giving us insights into how this disease is developing over time, even before the animals become symptomatic, even before there's any sign of malignancy in there. I think the time frame with which these animals are giving us or yielding full-blown mesothelioma really enables us to rapidly expand our mechanistic basis or mechanistic understanding of how this disease develops over time. And moreover, it also enables us to de-risk biomarker validation. So when we find molecular features in our mice that resemble or are identical to the molecular features that we're finding in human disease, that gives us confidence that we really have hit upon something that is intrinsically part of mesothelioma, and that makes it easier to narrow down what should we be looking for as these patients, in patients that present with potentially benign disease that may or may not be malignant. So can we do a better job of defining indicators that a person has mesothelioma even before we can find invasion? And so some of the specific questions that we're now trying to address in my group, it really come down to this question of diagnosis. And I think one of the great challenges with mesothelioma, the problem that we have right now is the diagnosis of mesothelioma currently requires finding cells that have invaded into the underlying tissue. And the problem, of course, with mesothelioma is that unlike many other cancers, it doesn't grow as a solid mass. It doesn't grow as one sphere. It grows as a field. And so even using scope-guided multi-region sampling and taking multiple biopsies from multiple regions, there's no guarantee that the clinicians are actually going to recover an area that shows invasion, and so that they're left with a potentially a suspected case of mesothelioma, but they're not able to give a definitive diagnosis. And so to really get down to the nitty gritty, if you consider a case study here, a patient that presents with CT scans showing pleural plaques, they have asbestos-related pleural effusion, fluid in the lung. Even now, immunohistochemical measures of known genetic lesions, such as loss of BAP1, such as loss of P16 or CDKN2A, as the gene is called, if there's no evidence of invasion, the clinicians still can't call this mesothelioma. And the situation that they find themselves in is that the patient goes on a wait and watch strategy. There's no intervention. And so if we can do a better job of using our mouse models and combining that with longitudinal analysis of human patient samples, perhaps we can define better diagnostic tools that will enable the clinicians to give a definitive diagnosis earlier. And so the question that sort of jumped out at us is, how do you survey the entire pleural space rather than going after a small number of discrete regions for biopsies? How do you really look at the entire pleural space? And so we sat down and thought about what are some of the features that we would want in order to be able to do this. And so, in thinking about this, whatever it is we're going to use should be small and it should be recoverable. We should be able to get it back out of the patient. Whatever we use should be able to detect and measure a broad spectrum of molecular elements, whether it's proteins or metabolites or what have you, because we don't know what molecules are actually going to tell us that are going to inform us of whether or not a patient has mesothelioma. Whatever we use would need to have very high sensitivity. Would need to be able to pick up really minute quantities, tiny fractions of a gram of any of these materials that may be informing that there's mesothelioma somewhere in the chest cavity that hasn't been picked up by biopsy. And we want ultimately a stable, reproducible readout. Well, it turns out that that's precisely what white blood cells do. So you get lots of white blood cells getting into the pleural cavity, surveying the entire pleural space, and they're incredibly sensitive to the kinds of molecules that are being produced by the environment. And they respond to those molecules by changing their gene expression patterns. And so considering various disease states here, either chronically inflamed mesothelium that's benign, truly benign, or indeed pre-malignant inflamed mesothelium, where you may have one or two malignant or a small number of malignant cells, and currently we're unable to distinguish between those two disease states. And then on top of that, going the full distance to full-blown mesothelioma. So these three disease states are all going to produce a different pattern of molecules that they express and they secrete into the pleural space that have the potential to alter white blood cell gene expression. And so our plan moving forward, and this is a grant that has recently been funded by the British Lung Foundation, now Asthma UK British Lung Foundation, we call it DEBIT-MESO. And the idea here, DEBIT-MESO, stands for the differential expression of bystander transcriptomes for diagnosis of malignant pleural mesothelioma. And the plan is very simply to make use of this pleural effusion fluid, this fluid that collects in the patient's cavities and currently gets poured down the drain and discarded and isn't used for any biological purpose. And so through two ongoing clinical trials, ASSESS-MESO, which Anna Bibi is going to tell you about as soon as I conclude, and also Meso-Origins. So ASSESS-MESO is going to give us pleural effusion samples from patients with confirmed mesothelioma, whereas Meso-Origins is prospectively sampling patients that are at risk of developing mesothelioma but have already presented with benign pleural effusion. And so the plan is very simple, that we're going to take these pleural effusions, we'll put them through a centrifuge to separate the cell pellet from the supernatant, and then we're going to put those fractions into a really deep molecular omic analysis. The cell pellet will go through an RNA-seq protocol, giving us gene expression information of what genes are being expressed within the populations of cells that are in the pleural cavity. The supernatant, we can apply mass spectrometry analysis to the supernatant to look at the proteome, to look at the metabolome. And ultimately, what we're hoping to find from these analyses, the expected outputs are that we'll get differential gene expression signatures in the white blood cells that are indicative that there is a cult mesothelioma somewhere in the pleural cavity of these patients, or alternatively that they are predictive of progression to full-blown mesothelioma. We're additionally going to get identification of the regulatory molecules and the secreted metabolites that control these white blood cell phenotypes. And this potentially gives us opportunities for intervention, but we'll see exactly how far that goes. Under a second study, IMED-MESO, for which we are hoping to get funding, we've applied to CR UK and will hear any day now if they're going to support this continued work in mesothelioma. We're going to combine the outputs from DEBIT-MESO with artificial intelligence-guided analysis of paired biopsies. These will be biopsies that were taken retrospectively from patients that went on to develop mesothelioma. We'll go back to their initial biopsies and ask, what did we miss there? We're also going to have the potential to re-biopsy very brave volunteers who have stable benign disease over a 2-year period. And ultimately, we're going to combine that with parallel analysis in our genetically engineered mouse models, where the key question here is, in mice that are destined to get mesothelioma, where we know that if we leave them long enough, they will get meso, we can go back and sample their disease progression early and look at pre-invasive disease states. Can we distinguish molecular features that separate distinguish that pre-invasive disease state from non-invasive disease in an animal that's at very low risk from developing mesothelioma? And it is through the cross-species conservation that we hope to filter and focus on high confidence discriminating molecular features for better diagnosis of mesothelioma. I'm over time and I apologize for taking so long. I'll quickly acknowledge my 4 lab members that have worked on building these mouse models and have a fabulous team. I'm so proud of all of them. I also want to acknowledge my key collaborators, Marion McFarlane and indeed Kevin Blythe, Anna Bibi also for her help and her willingness to provide us with pleural effusion samples. I need to acknowledge funding support from the British Lung Foundation, who have been stalwart supporters of our lab's work for the past 10 years, either in association with the McKnighton Mesothelioma Research Fund, with the Scottish Government Chief Scientist Office, or indeed now currently with Asthma UK. I want to thank Cancer Research UK and indeed the June Hancock Mesothelioma Research Fund. And last but not least, to acknowledge our patient and public advocates, Julie Roberts, Angela Elliott and Karen Blythe, who give us the patient experience and give us the experience of people who have lost a loved one to mesothelioma. And I think it's incredibly important to help us focus our research on topics that matter. So I'll stop there. Thank you.